In your book, you will come to a section that is entitled Important Principles That Can Characterize Single Gene Diseases. If I had written this book, this section would be entitled Things That Make It a Whole Lot More Complicated and Mess You Up, because the rest of this chapter involves complications, things that do not fit the typical patterns, things that don't fit the usual rules that we talked about at the beginning of this chapter. But I want to be sure that you know what to do on your exam. On your exam, if they simply show you a pedigree and ask for the mode of inheritance, then you should forget about all of these complications and only apply the basic rules that we used in the first part of the chapter. If on the exam, if they want to ask you about one of these complications like incomplete penetrance or anticipation or imprinting or any of the things in this list, they will call your attention to that complication in your exam question so that you will know then that they are asking a question about the complication. So don't bring the complications into your analysis of a test question unless the question writers tell you to, essentially. Now, there are a long list of complications here: variable expression, incomplete penetrance, and so forth. We're going to cover all of these just as they occur in your book. The first example of a complication is variable expression. Variable expression simply means that not everyone with a certain genetic disease will have the same severity of symptoms. That is, for any given genetic disease, there will be some patients who have very severe symptoms of that disease, whereas there will be other patients who have much milder symptoms of that disease. Well, how can we explain that variability in the severity of symptoms? Sometimes that can be explained by the kind of mutation that a particular patient has. For example, as we mentioned earlier, we might have a patient who has a particular genetic disease caused by a missense mutation. Now that patient, because the patient has a missense mutation, the patient might still have partial enzyme activity, might not have a complete deficiency of the enzyme in question, and that patient is likely to have milder symptoms of the disease. On the other hand, you might have a patient who has a nonsense mutation or a frameshift mutation causing his disease, and that patient is more likely to have complete enzyme deficiencies and more severe symptoms. So sometimes the particular mutation involved can explain variable expression. There are other times, though, when we simply have no idea why there is a variation in severity of symptoms for particular patients. The example your book uses for variable expression is hereditary hemochromatosis. This is a disease in which the patients accumulate excess levels of iron in their bodies, and that excess iron accumulates to high levels and is deposited in tissues all over the body. Hereditary hemochromatosis is an autosomal recessive disease. These patients typically have a wide variety of different kinds of symptoms because the iron is deposited all over their bodies. They typically will have arthritis because of iron deposits in their joints. They may have heart disease because of iron deposits in their heart tissue. They may have diabetes because of iron deposits in the pancreas. Their skin is sometimes a bronze color because of iron deposits in the skin. Sometimes people refer to this disease as the bronze diabetes. Well, the severity of those symptoms in each patient will depend on how much excess iron has accumulated and how much has been deposited in the tissues for that individual patient. The amount of excess iron that accumulates will depend on the amount of iron taken in during that person's lifetime and the amount of iron that the person gets rid of during the person's lifetime. The amount of iron taken in will depend on the food you eat, the water you drink, whether or not you take iron supplements. The amount of iron that you get rid of in your lifetime depends mostly on whether you're male or female. Females get rid of a lot of excess iron by menstruation, pregnancy, lactation, and those things all help to protect females from the severe symptoms of this disease. Or on the other hand, you might have a male who's a really good citizen and goes down to the Red Cross and donates a unit of blood every 56 days the way they want you to. By making all those blood donations in his lifetime, he would be accidentally protecting himself from the severe symptoms of this disease. So hereditary hemochromatosis is an example of a disease showing variability in the severity of symptoms, variable expression. Another example of a disease showing variable expression is neurofibromatosis type 1. Neurofibromatosis type 1 is a disease, an autosomal dominant disease, in which the patients develop benign tumors at nerve endings. Each of the little bumps on this person is a benign tumor at a nerve ending. Now, this disease shows variable expression because at the extremes, there are some patients who have this disease who have such a small number of these tumors of such a small size that those people don't even know that they have the disease. And at the other extreme, there are patients who have extremely large tumors from this disease. So very, very variable expression, differences in the number and sizes of the tumors that are produced. With variable expression, all patients have symptoms. It is the severity of symptoms that varies from patient to patient. Another complication is incomplete penetrance. On your screen it says, some individuals who have the disease genotype do not display the disease phenotype. What this means is, for certain diseases, when you are analyzing a pedigree or working with a family in the clinic, there are some times for certain diseases when you will be able to identify a particular person who you know has the mutation. That is, you know that this particular person has the genotype that says that he or she is supposed to have the disease, but that person will be completely normal. The person will have no symptoms whatsoever. That would be an example of incomplete penetrance. Patients that show diseases showing incomplete penetrance are usually diseases in which there is some sort of secondary event required to cause expression of the symptoms. That is, simply inheriting the mutation is not enough. In addition to inheriting the mutation, something else must happen in order to lead to the production of the disease. For these diseases, we refer to the degree of penetrance of the mutation, the proportion of obligate carriers who display the disease phenotype. What that means is, of all those people who inherit the mutation, what fraction or what percentage of them go on to develop the symptoms of the disease? In just a minute, I'm going to tell you about a disease called retinoblastoma, inherited retinoblastoma. And I will tell you that inherited retinoblastoma is 90% penetrant. What that means is, if an individual inherits the mutation, that person has a 90% chance of developing the disease, retinoblastoma, but the person also has a 10% chance of being lucky and not developing the symptoms of the disease. Here's the example of the pedigree in your book showing an example of incomplete penetrance for an autosomal dominant disease. I like to use inherited retinoblastoma as an example that fits this pedigree. Inherited retinoblastoma is cancer of the developing retina. It's caused by mutations, inherited mutations in the RB gene. The RB gene is a tumor suppressor gene. Now, inherited retinoblastoma is inherited in an autosomal dominant fashion. And that is a fact, and I'm telling you that fact. Now that you know that this is an autosomal dominant disease, what is wrong with the pedigree? The problem with this pedigree is that this male here, with the arrow pointing to him, this male does not have the disease. That is, the disease has skipped this generation in this branch of the family. This is autosomal dominant. We would not have expected to find a skipped generation. So inherited retinoblastoma is an example of a disease showing incomplete penetrance. That is, this male has the mutation but does not show the symptoms of the disease. We know that he must have the mutation because he has inherited it from his father, and he has passed this mutation along to his two sons. So he must have a copy of the mutation, but he does not show the symptoms of the disease. It's incomplete penetrance. How can we explain that? For inherited retinoblastoma, this is the explanation. The disease is caused by inherited mutations in the RB gene, which is a tumor suppressor gene. Now, one of the basic facts about tumor suppressor genes and their relationship to the development of cancer is that in order for a cell to become cancerous, we must have mutations in both copies of the same tumor suppressor gene. Both copies of the RB gene must be mutated in an individual cell in order to lead to the development of the tumor. What is being inherited in this family is one mutant copy of the RB gene. So each of the people in this family with the disease, as well as this man with the arrow pointing to him who does not have the disease, each of those people is heterozygous. They all have inherited just one copy, one mutant copy of the RB gene. In order for the tumor to appear, the patient must acquire a secondary mutation in just one single cell of the developing retina, so that one single cell is homozygous